What you are going to be doing today is building molecules that look like these. And you're going to be using these twisty little foam, they're like foam-covered wire. Whoever makes them calls them tubers because they're kind of like tubes. You're going to be going through all of these steps, so it's going to be real clear what this means. Basically, very last line on this last page. Right. Hydrophilic means a chemical that likes water. It's the very, very last line on the previous page. Things that are hydrophilic dissolve in water. They like water. They're not repelled by water. And sugar is hydrophilic. And things that are hydrophobic hate water. They do not like water. They're like oil and wax. They are repelled by water and they don't want to be anywhere near it if they can help it. The parts of a protein that you're going to be building this out of, you're going to see are either hydrophilic or hydrophobic, or there's a couple that are in the middle. And because your cells are full of water and surrounded by water, water is going to affect what these proteins do. Parts of the protein are going to want to get away from water. You'll see it real clear when we do this today. Parts are going to try and bend so that they're facing the water. All that this means is proteins fold into crazy three-dimensional shapes. There are 4 what they call levels of structure. When you write primary, the abbreviation looks like 1 degree. Secondary looks like 2 degree. Tertiary and quaternary, 4th level. Right, that's just like the shorthand abbreviation for primary, secondary, tertiary. So if you see that, that's what it means. All right, when we took a look at those pictures of those little pop beads, the order you strung them together is what made each one different. If you had 5 of those little colored plastic beads, if you had red, red, blue, red, yellow, that's different than blue, yellow, green. Same plastic thingies, but it's a different combination. The order, the way you could read them, blue, blue, green, yellow, blue, whatever, that's called the primary structure. It's the sequence of amino acids. It's the order of the monomers. And I'm not even sure if that's a word, but it's writable. Like you can write out histidine, glycine, isoleucine, threonine, proline, histidine, whatever. You can write out in like abbreviations or whole words if you're going there, like the order that the amino acids are.
He said somebody was looking
Mr. Bach had it?
Yeah, he said somebody was looking at it. So
Told you. By the way, Taylor, yeah, sure, you can go check and see where it is.
Oh, I apologize.
Yeah, right, you do. We know you. All right. Now, we're not going to talk about this secondary structure too much, but because of the way that these little amino acids attract each other or repel each other or attracted to water or repelled by water, there are 2 shapes that sections of the protein can take. We're not going to do anything with it. All I need you to realize is that when you see like the 3-dimensional shape of a protein, you will see sections where the protein is twisted into this little corkscrew, this little curlicue. That is this shape. A helix means it's, when you say it out loud, it's alpha helix. It's this little corkscrew shape. That corkscrew shape helps hold the protein in its shape. It stabilizes it. So both of these have the same corpus. Right? The other one is this zigzag pattern, which is the B stands for beta. You're not really going to get asked about this much, except for knowing that the purpose of both of them helps to stabilize the shape of the protein. That's about all you're ever going to get asked about those. So, if it asks, which of the following is something that helps to stabilize the shape of a protein, alpha helix and beta sheet could be answers. What we care about is this one. Tertiary structure is the one that matters. I showed you that little poster the other day. Okay, so the structure of something determines how it works. So let me ask you a question. Janesia, which one of these things up here do you think makes your hair? Well, yeah, that's its name, but just based on what they look like, which ones do you think would be reasonable in hair? I think it's these guys. So, but the idea is they're long and stringy. You don't build long, stringy body parts out of little round, nubby things. You build them out of, if you look at a rope is made out of long strands all braided together. It's not made out of little beads all glued together. That just wouldn't make any sense. If you're talking about something, though, that needs to be able to move around your body easily, then that's why you want things like this. They are tiny. They can fit through small openings. They're easy to circulate. This can't. This is just absurdly huge. This is too big for something that you want to be able to try and move around your body. So the shape that these proteins take is what makes them work the way they do. And screwdrivers and hammers are both made out of metal. It's the shape of the metal that makes them do one job or the other. This three-dimensional shape is what determines the function or purpose of the protein. If the protein loses its shape, it stops working. All right. Every protein has these first three levels of structure. Every protein has individual amino acids strung together in an order. Every one of them will have little sections that either twist up into corkscrews or fold into little beta sheets. And every one will fold into a big three-dimensional pretzel, but only a few will have what's called quaternary structure. Quaternary structure is where more than one whole protein sticks together into like a big super multi-part protein. A good example is the hemoglobin that makes your blood red and carries oxygen. it's made of 4 separate proteins that stick together into a supergiant protein. Each of the 4 pieces gets made separately. Each of them is its own polypeptide protein. But for these things, they only work if you have all 4 pieces. Most proteins don't form like big multi-part things. Alright, so since shape is what matters, losing the shape is what destroys a protein. Denaturing is what you call it if for some reason a protein unravels and loses its 3-dimensional shape. I'm not talking about digesting it back into amino acids and like making it gone, but there are things that can take a protein and it doesn't fall apart, but it unravels into like just a big long ribbon of protein again. Denaturing destroys the function of that protein. If the shape of a protein is what makes it do what it does, if you make it lose its shape, it doesn't do it anymore. I mean, you know, if you have some kind of thing made out of plastic and you melt it down, it's still plastic, but it doesn't do what it's supposed to anymore because it's not the right shape anymore. You made it useless. The most common things that will do this inside of a living thing are heat and changes in how acidic things are. Normal body temperature is about 98, which is about 37 degrees Celsius. What is the highest fever you have ever heard of anybody getting?
103.
Oh, that's just feeling crappy. Like 106, and you're probably completely loopy, talking nonsense, can't stand up. Right? Basically, your body can handle up to about 108 degrees. At 108 degrees, proteins inside of your brain cells start denaturing. They used to do something to keep your brain cells working, and now they're falling apart, which means whatever they used to do, they're not. That's the reason why as your fever starts to get higher, people get a little weird. They start like hallucinating, they start talking nonsense, they get dizzy, they can't stand up, because they're losing the proteins that keep their brain cells working. You're going to find that almost always the first thing to go in your body is brain cells. That's because they have the most needs, they use the most energy, and they operate the fastest. So if anything slows your body down, the fastest working part is the part that feels it first. If you run out of oxygen, because your brain cells run faster than all your other cells, they use up their oxygen before the rest of you. That's why if you like fall asleep and your arm falls asleep and goes numb, anybody ever wake up and have their arm so asleep they can't even move their hand? Like it's like just like limp. And then you move, the blood starts going, and then you can move again, and then the pins and needles come. That's because you cut off blood to your arm. Everything else was fine, but your nerves fell asleep because they used their energy faster than everything else. So when they got like their oxygen supply cut off when you laid on your arm funny, they basically passed out and stopped working. And so everything else was fine. It's not like your arm is going to die, but the nerves stopped, so all the messages stop. When you get the blood going again, that pins and needles feeling is the nerves all waking up like in random order, and as they all wake up, they send a message back that says, whoa, I'm here. And the problem is it's happening all over in no particular order, and you're feeling all the nerves just waking up all over your arm, and you think it feels like things just touching you everywhere as the nerves just randomly wake up all over the place. Luckily, in you, if you can cool somebody back down, all of those proteins will coil back up again and start working because it's reversible.
Even the ones in the brain.
Yes, but you can't have gone too long. If somebody's got a fever of 107, you need to get like ice on this person. You need to get them in like a cold bath or something, because the longer they have a fever of 107, the longer the proteins aren't working, the longer your brain cells aren't getting what they need, and eventually the cells are just going to actually die from the lack of whatever those proteins were supposed to do. Once the cells die, then cooling them back down doesn't work anymore. But if you can cool them back down before you actually killed brain cells, and if you have a fever of 107, you're killing brain cells. So if you can cool them back down again, then that's supposed to say reverse. It's reversible. You heat something up enough, and you will denature the proteins so much that they won't go back. That's what you see when you cook food. Non-reversible is what explains all those color changes in food that you cook. It's why egg whites go from clear to white, and they don't go back again when you stick them in the fridge. Meat all has those different color changes they go through. That's because the proteins they were made out of got so hot, they denatured so much that now they can't coil back up again. Right? And so they're permanently stuck that way. This is not something that happens in people or any other living thing unless you actually make it so hot that you cook it. And I don't know what color people turn when they're properly cooked. I don't know what cooked people look like. I don't know what color human turns. You know, it's like, you know, lobsters go from brown to like, you know, that kind of red, pink lobster color. I don't know what color we turn when we're cooked right. But pH can do it too. Changes in how much acid there is, and in fact, you've got even less tolerance for that. Your pH is something like, I think it's like 7.34. Remember, 7 is like neutral. 7 is just water. So 7.34, is that slightly acid or slightly base? Acid would be like a 2. Base would be like a 10. So we are just slightly not acidic, slightly basic. Your body can literally only tolerate about 7.55 down to 7.2. It's like a really skinny band that your body can tolerate in terms of like acid buildup. Your kidneys do an awesome job making sure that doesn't happen. For example, if urine starts to build up in your blood, it's acidic. And so you pee it out and it gets rid of that acidity and gets back. Your kidneys do all kinds of stuff to adjust that, but the same thing happens. Proteins start denaturing, they stop working. Okay. All of these functions that proteins do are kind of self-explanatory the way they're listed. I'm going to make sure I've pointed out at least one example for each one. On the AP test, you will almost certainly get a question that says, explain the functions of blah, 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 and give 2 examples. I'm going to constantly give you things that, okay, they're not the only example, but at least you'll have an example. This one's already listed. Structure, keratin that makes up your hair, your skin, your fingernails, and this is the most numerous protein in your entire stinking body, collagen. It is everywhere. It's what holds your skin onto your body. It's what holds all of your muscles onto each other. It's what holds your muscles onto your bones. It holds your bones to your other bones. It reinforces the insides of your bones. It wraps all of the muscles. It reinforces the walls of your arteries so they don't pop when your blood pressure goes up. It's everywhere. But it doesn't do anything. Like they don't have a function like that's like saying, well, what's the function of the chair legs on your desk? Yeah, it holds you off the floor. What are they doing? Nothing. They're just there in the way so you can't hit the floor. It's not like they're actively using energy and pushing up on you or something. They're just physically, structurally there. And so, what does it do in your skin? Well, it doesn't do anything. It's just laying there being a really good waterproof barrier so that the outsides of you doesn't look like the inside of your mouth. Protein storage. Albumin is one. Albumin, for example, in an egg, the reason that the egg white is there is so that the growing chicken has protein. Inside of your body, you've got a protein called ferritin. Any idea what it stores? Iron. Oops. If you remember, Fe is the chemical symbol for iron. Right. So you've got ferritin that stores iron. Transport. Hemoglobin carries oxygen around your body. Oxygen does dissolve in water, and in theory, if you just had blood made of water, it would circulate some oxygen, but not enough. Oxygen doesn't dissolve in water well enough that it would do you a whole lot of good. So almost every living thing has developed some kind of protein in their blood. Ours uses iron and is red. There are other organisms that use other types of things. There's like horseshoe crabs use, I think, either copper or cobalt, and their blood is bright blue. Does the same thing, it's just nature came up with a different way of doing it for them. Hormones. Hormones are things that are chemical messengers in your body. We're going to do a whole unit on them. Their job is to communicate with parts of your body to tell them to do something. Not all of your hormones are proteins. For example, estrogen and testosterone are not proteins. Those are actually made out of cholesterol. So not all hormones are proteins, but a lot of them are. Receptors are the structures on a cell that detect hormones. You can't tell a cell to do something if the cell has no way of knowing that the hormone is there. You release insulin to tell cells to do something. The cells have to have insulin receptors so that they notice that the insulin's there. We are going to talk about, well, basically these two go together. These two are basically the two halves of one job. I mean, you can't talk to somebody if they don't have the ability to hear. It's like two halves of the same purpose. Yeah.
Can't operate without the other.
Right. Well, I mean, sending hormones out to cells that don't have receptors has no purpose because they can't hear the hormone. And cells that have receptors but there's no hormone means, like, in essence, you can hear but nobody's talking to you. So still nothing gets done. Movement. There's a protein called dynin that is what waves the flagella back and forth on, well, yes, unicellular swimming organisms, or in humans, sperm. And there's a protein called dynin that waves the flagella back and forth and makes it swim like it's swimming with its little tail. Also, all of your muscles move by two different proteins. Where's my poster? These two. This red one and this blue one are layered like this. So it's like a blue, red, blue, red, blue, red, blue. And they pull on each other, and that's what's going to make your muscles contract when they pull together. And then when you straighten an arm back, a muscle back out, it pulls it back out. And then it stops, and so when you're stretching, it's when you're trying to get it to pull even further than it usually normally would. And if you tear a muscle, it's because you pulled so hard you went, and now they aren't touching anymore, and so now they can't pull anymore. Which is why if you ever really pull a muscle, yeah, you'll be really black and blue because you ripped a lot of blood vessels, and wherever you pulled that muscle, those proteins don't touch anymore, and so that muscle is going to end up being like weaker in that spot because that spot doesn't function the way it's supposed to. Defense. All of the antibodies in your bloodstream are all proteins. The ones that fight off infections, the ones that you build up when you get a vaccine. An enzyme carry out metabolic processes. Basically, there's virtually nothing that happens in a cell that an enzyme is not doing. Any chemical reaction that happens in a cell is being done by an enzyme. The majority of your proteins are enzymes. And when we look at this poster, most of the thing on this page are enzymes. Right? Like all of these things, well actually all of these are enzymes. They're just showing you a group, but all these are enzymes and the majority of them are enzymes. Okay, one page left. And we're going to do this real fast because we're doing an entire chapter on just the cell membrane, so we're not going to get too deep on this. What? Okay, lipids. So far, everything we've been talking about was a polymer. Lots of little pieces strung together in a big long chain. Lipids are huge, but they're not polymers. They're not a big chain of things. Basically, they come in one of two shapes. Lipids either look like a ginormous stretched out letter E, or they look like a ginormous stretched out letter F. They either have three big, really long tails, or they've got two. But that's their shape. You don't have like then another letter E strung onto there and another onto there. It doesn't do that. They're just one molecule. It's big, but it's way bigger than an amino acid, but they're just individuals. They're made of two parts. The big, long, skinny sections are called fatty acids. And then the little, like, backbone of them are called glycerol. So in the case of a letter E, it's three fatty acids connected together on one glycerol. The one that's the letter F is two fatty acids connected to a glycerol. If you measure somebody's, you're good? No. What you're going to do is you're going to take a picture of it with your phone and upload it to Schoology. There's a an assignment in Schoology that says upload your photo of the crime scene sketch. Right, and then that's it. Lost my train of thought. Oh, triglycerides. Triglycerides are what you call like your lipids. If somebody has like high lipids in their blood, it's high triglycerides. It's the stuff that's in like fat. Basically, the characteristic of these things is they are very, very, very non-polar.
What the lipids are?
Yeah, well, because what you've got is you've got 3 giant fatty acid chains that are super non-polar and a little tiny speck that is a little bit polar, and overall this thing hates water with a passion. This is literally the definition of hydrophobic. It's oil. You know how well oil mixes with water. It's wax. You know how well wax mixes with water. It doesn't. That means what we tend to use these things for is for either storing energy because of how big they are, but the one that we're going to talk about the most is this. This is your cell membrane. Every single membrane, the cell membrane, the nuclear membrane, the membrane that makes every vacuole and Golgi body and endoplasmic reticulum and mitochondria and all of those little compartments in the cell are basically made out of what would amount to fat bubbles. You probably remember doing the bubble lab last year in bio where you blew those bubbles and then you tried to stick the tube through them. And structure-wise, soap bubbles have the exact same shape as membranes, except that these are molecules of soap instead of molecules of lipids that we use. We're not made of soap. These guys that we make membranes out of are a specific type called phospholipids. The phospho part refers to this yellow thingy up here, which in the middle of it is a little P because that's a phosphorus. That's an addition that you only see on these F-shaped ones, and it makes them weird. It makes one side of the molecule that they always draw like a ball very polar, like it loves water. And then the 2 long stringy parts that make like the things on the F are very not polar. There's a word for this. A molecule that is polar and not polar at the same time is called amphipathic. And it's going to give these things the perfect characteristics to make your cell membranes. It's really wet inside you. There's water outside the cell. There's water inside the cell. These things line up like this. They can stand being in water because the parts that like water are facing in and out where the water is. We'll go over this way more when we do the chapter 5. And the parts that hate water are in the inside. What this means is all the membranes in your cells like water enough that they can be surrounded and filled with it and it won't like freak them out, but they can't be dissolved by water. If you made cell membranes out of carbohydrates, they would dissolve. If you made them out of protein, they would dissolve. if you make them out of this, the water can't dissolve it because the middle doesn't want anything to do with water and it sticks together. But it's not just, you could do that with something that's totally non-polar, but it would hate water all the time. So this is the perfect combination of, it can stand being around water because it has the polar end, but it won't get dissolved by water because it's got the non-polar end, and that's what life needed to make the membranes that we're built out of. Things that would hold together in all that water, but could stand being surrounded by it at the same time. Okay,